Good afternoon one and all. This is your Prasanna ma'am from Chandu Biology Classes. So yes, so far we have completed 2 chapters from our PGT Biology syllabus, and today we are going to start the 3rd chapter, that is Microbial world, wherein we are going to learn in detail about virus, bacteria, algae, fungi and protozoans, all the unicellular organisms, be it prokaryotic or eukaryotic. And most important thing is we have to look into the useful and harmful microbes in detail. So all the useful and harmful microorganisms, complete life cycle and everything, general characteristic features, types of them, everything we are going to exercise in this chapter, including the objective. So let's begin with the introduction into the microbial world. What is microbiology? Microbes means small, bios means life, logos means steady. This term has been originated from Greek, okay, microbiology, which is originated from Greek, which says they are simple, the study of simple small life organisms is called as microbiology, okay, which is one branch of biology. This deals with the scientific study of microorganisms. This includes protozoans, which are animal-like protists, microscopic algae, which are photoautotrophic, fungi like yeast, Saccharomyces cerevisiae, and molds, slime molds, which are again fungi-like protists, and bacteria and viruses. Viruses, of course, they are crystalline entities or living entities. They are not said to be the living organisms, but then we are going to learn viruses even in this chapter. So all the prokaryotic and eukaryotic microscopic organisms are learnt, or unicellular organisms are learnt in this chapter. So microbes are omnipresent in vast numbers. They are omnipresent, ubiquitous or cosmopolitan in distribution. They are present everywhere in all the habitats. So what is the historical contribution in discovery of viruses? Whenever we talk about the discovery of viruses, the very first name which strike our mind is Anton van Leeuwenhoek, whose tenure is from 1632 to 1723. He has pioneered early work on microorganisms. And then Louis Pasteur from 1822 to 1893, this is his tenure, where he developed pasteurization and vaccination techniques, okay. Pasteurization and vaccination techniques were developed by Louis Pasteur. And then Robert Koch, he has contributed foundational work in microbiology. So the 3 eminent scientists primarily who has delivered their contribution in microbiology is Anton van Leeuwenhoek, Louis Pasteur and Robert Koch. So yes, now let us begin the study of microorganisms from viruses. Just a second. Yes. So viruses, they are best defined as the infectious particles, okay. Virus are best defined as infectious particles. Usually viruses are mostly having one proteinaceous envelope and the genetic material. But few of the viruses like herpes virus or HIV, they will be having an envelope also. So viruses will be having 3 components in it: envelope, and the protein called as capsid, and genetic material. Genetic material can either be DNA or RNA, okay. Those viruses which affect most of the plant cells, that is DNA, animal cells will be, plant cells will be RNA, animal cells will be DNA. So most of them will just have this proteinaceous code and genetic material. Few of them will also get encapsulated with envelope over the protein coat, okay. So now you see virus is best described as what? Infectious particles, and the infectious particle is the genetic material, not the protein coat. Each virus contains a single type of nucleic acid, either DNA or RNA, but not both the genetic materials, okay. Take a note of it. Nucleic acids is enclosed in protein coat, sometimes also covered by envelope of lipids, proteins and carbohydrates. So, the nucleic acid, that is genetic material, is uncovered by what? Protein coat. Sometimes it can also be the envelope of what? Lipids, proteins and carbohydrates. Mostly it is the protein coat, sometimes it can be these components also in the envelope. Next, viruses are obligate intracellular parasites. Okay. So, they are most well noted as what? Obligate intracellular parasites, and they cannot reproduce unless they invade a specific host cell. So, virus cannot replicate outside the host cell, they can replicate and increase the machinery only when they infect the host cell, only within the host cell. They use host genetic material and metabolic machinery to produce their progeny. They cause serious damage and disease in the humans, plants and animals. This scientific study of virus is called as virology or virology. This is the basic description, fundamental description of viruses. And then here you see, viruses do not exhibit most life processes of a cell. However, they maintain genetic continuity through multiplication. Okay. They will maintain that genetic continuity through what? Multiplication. Multiplication within the host cells. Okay. Once the entire multiplication is done through either lytic cycle or lysogenic cycle, then they will be dispersed into the environment. They undergo mutations, making them more than just inert molecules. Okay. They will undergo what? Mutations, and they will make them just an inert molecules. Now, let us focus or target on the discovery of viruses. Many a times the questions are asked from the discovery of viruses. First, we have to begin with the scientist Dmitry Ivanovsky in the year 1892. He is the first scientist, he is the Russian pathologist who is working or who has worked on tobacco mosaic disease in tobacco leaves. Okay. What did he do? He has used filtered sap of tobacco leaves. The filter was designed to trap the bacteria. Even after filtration, the disease symptoms appeared in the healthy plants. Okay. So, what did he do? He has taken an infected tobacco leaf. Okay. He has grinded it with mortar and pestle. He has extracted the sap and passed it through the bacterial filter paper. But the sap, the infectious particles which are in the sap has passed down through the bacteria's filter paper. So, virus size is less than that of bacteria. So, they have passed out easily through the bacterial filter paper. So, when this sap is injected to a healthy plant, even this has shown the disease, which indicated that the infectious sap or the infectious particles has passed down through the bacterial filter paper. Okay. It didn't trap in the bacterial filter paper, it just passed down through it, because virus particles are less in their size when compared to the bacteria. He concluded that filterable agent caused the disease. That filterable agent which has passed down through the bacterial filter paper has caused the disease. So, this is the discovery part of Dmitry Ivanovsky. What did he do? He has worked out on tobacco mosaic disease. He has taken an infected tobacco mosaic plant, extracted the sap of it, passed it through the bacterial filter paper, and he has observed that the infectious particles are still present in the filtered sap. Okay. Next. Yes, Martinus Beijerinck. Okay, he is the next scientist who has repeated the Ivanovsky's experiment. And what did this scientist do? He has transferred that infectious sap, he has transferred this infectious sap into a healthy tobacco plant, Fine. He has transferred this infectious sap into a healthy tobacco plant, and he called this as contagious living fluid or contagium vivum fluidum, okay, wherein the infectious sap has caused the disease even in the healthy plant. That is why he named it as what? Contagium vivum fluidum, which is well noted as contagious living fluid. Till here it is clear, right ma? Next, W.M. Stanley. W.M. Stanley in the year 1935, he has purified this sap. He has purified this sap and crystallized. He has purified the sap and crystallized and named that as tobacco mosaic virus. So, who named tobacco mosaic virus at the end? W.M. Stanley. Who has purified the infectious sap? W.M. Stanley. Who has crystallized the virus? Crystal virus structure or crystalline virus structure is given by whom? W.M. Stanley. Remember, this format only we will get the questions. Next, it is Frankel Conrad in the year 1956. Okay, Frankel Conrad in the year 1956. He has confirmed that the genetic material in tobacco mosaic virus is RNA. Okay, Frankel Conrad has confirmed that the genetic material in TMV, tobacco mosaic virus, is what? RNA. Used advanced technique. He has used advanced techniques like ultracentrifugation, X-ray crystallography and electron microscopy to study the viruses and their structures. Okay. So, who has used the advanced techniques to study the virus and their structures? If this is the question, then our answer should be Frankel Conrad. Virus particle crystallization is done by Stanley. Advanced techniques were utilized by Frankel Conrad. Clear, right? Next. Classification of viruses. So, from International Committee on Taxonomy of Viruses, this laid to classify the viruses. This governs the classification and naming of viruses. Okay, ICTV will govern the classification and naming of virus. This uses three level hierarchical systems. First is what? Family, second is genus and third is species. Okay. So, first family ends with what? Suffix viridae. Genus ends with what suffix? Virus. Species has English descriptions, for example polio viruses. Okay. So, classification of virus is done by ICTV. This uses three level hierarchical systems: family, genus, species. Family ends with viridae, genus with virus and species with English descriptions, example polio virus. Clear, right? Now, based on the disease caused by the virus, for example, virus causing AIDS is named accordingly. Okay, virus causing AIDS is named accordingly as what? HIV classification, human immunodeficiency virus. As family, the family of HIV belongs to what? Retroviridae. Genus, lentivirus. Species, human immunodeficiency virus. Clear, right? So, this is all about ICTV, International Committee on Taxonomy of Viruses. And next, structure of viruses. We have different morphological structures of the viruses, like the size of the virus will range from 300 nanometers to 20 nanometers. 300 nanometers, for example, tobacco mosaic virus, 20 nanometers, for example, parvovirus. So, the comparable, this is comparable to small bacterial cells like mycoplasmas and ribosomes. So, viral particles are very small in structures. They are comparable with this bacterial cells like mycoplasma, which are wall-less bacteria and smallest living cells, and ribosomes, which are non-membrane bound organelles. So, viruses are classified based on their shape into first what? Helical virus, second polyhedral virus and third spherical virus, fourth is complex virus. First, helical virus, for example, tobacco mosaic virus, which is the helical virus. I already told you, most of the plant viruses will have what genetic material ma? RNA as their genetic material. So, TMV, tobacco mosaic virus, has single stranded RNA. This is a helical virus. We are clearly going to study the tobacco mosaic virus structure now. Polyhedral virus, best example is adenovirus. This has DNA as their genetic material, and above the protein coat, envelope is made up of glycoproteins, carbohydrates and proteins. The envelope of this virus will contain carbohydrates and proteins, that is glycoprotein. Next, spherical virus, best example is influenza virus. This also has RNA as their genetic material, single stranded RNA, and a proteinaceous called as capsid. Above this capsid, it has a membranous envelope made up of again glycoprotein. We have to remember which viruses are having only genetic material and proteinaceous code, and which viruses are having an envelope encapsulated over the proteinaceous code. We have to remember that. And next is what? Complex. Complex viruses. Best example is bacteriophage. Bacteriophage is the best example for complex virus, wherein bacteriophage, we all know this infects bacteria and its genetic material is DNA. Covering this DNA, it has the proteinaceous coat, that is capsid. Okay, it has the proteinaceous coat, that is capsid. And bacteriophage has a head, collar and tail. Head, collar and tail. And lastly, tail fibers. Clear, right ma? So, what all types of viruses we have? Helical virus, best example is TMV, whose genetic material is RNA and which is covered by a proteinaceous coat, that is capsid. Second type is polyhedral virus, whose best example is adenovirus. Genetic material is DNA covered with proteinaceous coat capsomere. Above that, it also has glycoprotein. Third type is spherical viruses. Best example is influenza virus. The genetic material it has is RNA and the proteinaceous coat is capsid, encapsulated by a membranous envelope which is made up of glycoprotein. Then complex viruses, best example is bacteriophage. This will infect the bacteria. It has the genetic material double stranded circular DNA uncovered by the proteinaceous coat capsid, and its structure will have a head, collar, tail, tail sheath and tail fibers. Clear, right? This is the complete morphological description of different types of viruses. So, here I have tabulated completely. Once you look into this table also, first type is helical virus. We have learned the example is TMB. How can you describe helical viruses? As long rods, they may be rigid or flexible. Best examples include rabies virus and tobacco mosaic virus. Second, polyhedral viruses. They are of many sided shapes. Best example includes herpes simplex virus and polio virus. Third, enveloped viruses. This viruses with capsid enclosed in an envelope. These are roughly spherical. Best examples include influenza virus and human immunodeficiency virus. Fourth type is complex viruses. These are complicated structures, especially those which are infecting bacteria. Bacteriophages, for example, T4 bacteriophage. Okay. So, this is completely about morphological types of viruses. Now, let us look into the basic components of virus also. Some viruses like measles, they will be having some viruses like measles, they will have the spike like projections which are glycoprotein projections. They help in host cell attachment. Okay. This spike like projections which are made up of glycoproteins, they help in attachment to the host cell. Spikes are crucial for virus entry by binding to the receptor on the host cell. Next, basic components of viruses. We have two components of viruses so far we have discussed. First, core. It is made up of nucleic acid. Core contains nucleic acid genetic material, be it either RNA or DNA, not both, which is covered with a protein coat. We call it as capsid. Capsid has proteinaceous units called as capsomeres. Capsid has proteinaceous units called as what? Capsomeres. Capsid is made up of repeating protein subunits. Okay, capsid is made up of repeating protein subunits called as capsomeres. The number and arrangement of capsomeres are virus specific. Okay, it is specific for every virus. The arrangement of capsomeres, proteinaceous subunits inside the capsid is specific to each virus. So, this is the basic description or the components of viruses. Here if we see genetic material in viruses, that it can be, mostly in virus it will be circular double stranded DNA only, quite at times it can also be linear double stranded DNA. So, the genetic material in viruses, types of nucleic acids if you see, double stranded DNA, single stranded DNA and single stranded RNA, double stranded DNA, single stranded DNA, double stranded RNA, single stranded RNA. Double stranded RNA is dynamic, it will not be stable at any place, because double stranded RNA will not be there mostly, you can consider single stranded RNA only. These three are different types of genetic material which we observe in different viruses. So, trends if you see, plant viruses will have single stranded RNA, animal viruses will have double stranded DNA, bacteriophages will have double stranded DNA. Some viruses if you take like HIV, human immunodeficiency virus, they have two identical RNA molecules, okay, they have two identical RNA molecules called as genomic copies. What will be the shape of nucleic acids? It can be either circular or linear. Two different shapes of nucleic acids are present. Most viruses will have a single nucleic acid molecule, most of them will have only one single nucleic acid molecule. Okay. So, this is about the genetic material or nucleic acid description which we see in the viruses. So, today we will look into two major types of viruses. One is tobacco mosaic virus and the second is bacteriophage. Lytic cycle and lysogenic cycle we will be looking in the next class. So, here if you see, first tobacco mosaic viruses. The size of tobacco mosaic virus is 300 nanometers long, it is 300 nanometers long and 18 nanometers in the diameter, 18 nanometers in diameter. Fine, it is 300 meters long and 18 nanometers in diameter. What is the molecular weight? Molecular weight is 39 into 10 to the power of 6 daltons. Okay. So, remember the length and width of tobacco mosaic virus. Length is 300 nanometers and the width is 18 nanometers in diameter. Molecular weight is 39 into 10 to the power of 6 daltons. What is the structure of TMB? It is having a proteinaceous coat, that is capsid, which is made up of 2130 protein subunits. Okay. So, it is made up of how many protein subunits? This capsid layer, complete capsid is made up of 2130 capsomeres or protein subunits. They are arranged in a helical symmetry and each protein subunit will have 158 amino acids. Each protein subunit will have 158, it has 158 amino acids. This contains, the core contains single stranded spirally coiled RNA. So, it has single stranded spirally coiled RNA which is made up of 6500 nucleotides. Okay. This has 6500 nucleotides. Okay. And central hollow is having 4 nanometers or 40 angstroms. This central hollow is having 4 nanometers or 40 angstroms. Here what we have to remember, each and every particular of this TMB is important for us. Length is 300 nanometers, width 18 nanometers in diameter, and the capsid proteinaceous coat will have how many subunits? 2130 capsomere subunits, which has what symmetry? Helical symmetry. And each protein subunit will have how many amino acids? 158 amino acids. This contains single stranded circular RNA this RNA will have how many nucleotides? 6400 nucleotides approximately. So, if we have to take the ratio in between the number of nucleotides and the protein subunits of TMB. So, the ratio of number of nucleotides is to protein subunits. So, number of nucleotides 6400 nucleotides, right, approximately, and proteinaceous subunits will be 2130 proteinaceous subunits. So, mostly it will be 3:1 in the ratio. The ratio will be 3:1. Clear, right? And it has a central hollow portion that is 40 angstroms or 4 nanometers. If the ratio is asked in the reverse way, ratio between the amount of ratio between the protein subunits and number of nucleotides will be 1:3. Okay. So, 3:1, 1:3. Remember that. This is the complete structural description of tobacco mosaic virus. Next, when it comes to bacteriophage. So, here you look into bacteriophage. Best example we are taking is T4 bacteriophage, which is popularly known as tadpole like structure because it resembles a tadpole. Okay. So, at the center the genetic material whichever is present, this is double stranded circular DNA. Double stranded circular DNA is the nucleic acid. It is covered by the proteinaceous coat that is capsid, and it has 4 regions. First is head region. Head is made up of proteinaceous subunit that is capsid, and then it has a collar and then a tail, then tail sheath and fibers. Okay. Head, collar, tail sheath and fibers. So, whenever this bacteriophage is infecting the bacteria, whenever bacteriophage is infecting the bacteria, okay, this tail fibers, at the tip of the tail fibers, it has the enzyme at the tip of the tail fibers. The enzyme which is present is lysozyme. Lysozyme enzyme will degrade the wall layer of the bacteria, and it will aid in the pass of the genetic material from the head passing through the collar and the tail into the bacterial genome or into the bacterial cell. This is how the genetic material is passed out, and the tail fibers have the enzyme, that is what, lysozyme, which can cleave the wall layer of the bacterial cell, that is peptidoglycan cell wall. So, here head contains DNA, collar, and tail includes sheath, base plate, this is the base plate, and pins, these are the pins, and tail fibers. Okay. So, what does tail of a bacteriophage include? Sheath, base plate, pins and the tail fibers. The main functionality of this bacteriophage, tail fibers attached to the host cell, and at the tips they have lysozyme enzyme. They will hydrolyze the cell wall of the bacteria, and it will ease the passage of the viral DNA, which you can call it as tail sheath will infect or inject the viral DNA into the host cell. This viral DNA will be injected into the host cell. This is the complete description of bacteriophage and TMB. In our next class, we will be looking into the lytic cycle, lysogenic cycle, examples, sexual recombination that is happening in virus, that is conjugation, transformation, transduction, and bacteria also will look completely. The structure, types of bacteria and physiological functionality of the bacteria also we will be looking into. Okay. So, that is all for today's class. Thank you.